If you're looking at pastured poultry, then yeah, backyard flocks in the air are probably going to be a bigger factor. But I would think if you have good biosecurity practices, it's maybe not so much of a deal for like indoor raised birds.
This is Dr. Phil Stair with Biosecurity Poultry Podcast. Interview Dr. Andrea Etter. So, Andrea, we were just chit-chatting a little bit. Just go ahead and tell the crowd a little bit about yourself, where you grew up and your background there, educational background.
Yeah, so perhaps unsurprisingly, given my topic today, I actually grew up in rural Wisconsin, homesteading. I like to joke my parents were the homesteaders before homesteading was as popular as it is now. And they did move from Chicago to rural Wisconsin when I was a small child and got all the animals, essentially. We had an old McDonald farm, including poultry of various types. And then I went to college, started nursing, wasn't for me, switched to biology, wanted to teach at college level biology, microbiology, and so I went to grad school and eventually ended up in food microbiology, which is my happy place.
That's good to know. And so go ahead and tell the crowd what your research was in for your PhD.
Yeah, so I started off, my doctoral advisor was Haley Oliver, and she hired me to do PFGE. So glad we're not doing that anymore. And internal in a premature stop codon analysis on a set of about 900 Listeria monocytogenes from retail delis. Pro tip, do not buy sliced at retail deli meat. It's just so much, it's much more risky than prepackaged stuff.
Okay.
And then as that project wrapped up, we got started. She had money to do some work on stress tolerance and salmonella, and then there was a giant outbreak of Salmonella Heidelberg that was occurring around this time, and we decided to pivot, get isolates from USDA inspection of those facilities and test them for stress tolerance because everybody was going, okay, there's got to be something special about these. They can't just be that lucky. And so I spent the last couple of years of my PhD on that, got some USDA NIFA predoctoral funding for that, and had a lot of fun with it. We did find that a number of the isolates, particularly ones from a strain that was associated with illness from rotisserie chicken, had pretty high heat tolerance, like 56 degrees Celsius stationary phase. There was like 10 to the third, 10 to the fourth, so like 1,000 to 10,000 cells sticking around at the end of an hour.
My goodness.
Not great. Yeah. Significantly higher than the reference strain for Heidelberg. We did some RNA sequencing, and baseline levels of a lot of their stress tolerance genes were a lot higher than the reference strain, kind of suggesting that they were like the bacterial preppers. They were ready, and so they could cope better with heat.
This passion, that's good. I'm glad you've enjoyed that. That's some areas that I've not dealt in my career. Mostly mine's been on live animal side. So you're in the food safety side, which is obviously very important. But it sounds like now your work there in Vermont, you're kind of bridging over to the live side a little bit. You want to tell us about what you're currently doing?
Yeah, so when I got this job at the University of Vermont, the research I proposed was an extension of that stress tolerance study and then some larger studies on persistence in Listeria monocytogenes, but we're an agricultural college, obviously, and the first year as part of my startup package, they said, here, we'll give you 10,000 from the agricultural experiment station, but you have to propose research that benefits Vermont. And I don't know why I chose to go this direction instead of like studying Listeria, because dairy. But I was reading in the news all this stuff about backyard poultry associated outbreaks, and the two areas, like my upbringing and my interest in salmonella, collided. I'm like, man, all these people are getting sick, but I can't find any data on how common salmonella actually is in the backyard poultry. There were a handful of studies, mostly in cities. Greater Boston area was the big one. and they weren't finding salmonella. It was like really rare, like less than 2%. And all these people are getting sick, and I'm like, this doesn't track. It there's got to be more to the story. And I'm looking around in Vermont, and everywhere I drive, I'm seeing chickens, and I'm hearing chickens in my neighborhood. Like, okay, this is kind of a great place to look into this a bit more. And so I proposed looking at backyard poultry for salmonella, and got that project started with an undergrad the first summer I was here. And we didn't find anything. So that was a little, we had got 28 people to agree to let us sample their flocks, and we did cloacal swabs, which uh, definitely not great if you're dealing with people who have chickens that are free ranging, because chasing chickens all over somebody's yard in 85-degree weather is not fun.
Right. Yep. Typical Mississippi where I am. It's
Yeah.
85 all the time.
Yeah, I'm from the north, so yeah, it's not fun. One place had honeybees as well, and trying to corral a chicken that had escaped from the pen while we were sampling, I got stung by a honeybee, and I'm like driving all around Vermont. It was great for learning Vermont geography. We didn't find any salmonella, and at the end of the summer, I was sort of baffled and a bit discouraged. 28 flocks later, there's no salmonella. Okay. I'm like, well, I should probably sample a bit more, but I don't know how long this project's going to last.
I was going to ask you what you moved on to then. If you go ahead, proceed. That's good.
I still figured there had to be salmonella out there, and I just must have somehow missed it that year. And in January of 2020, so my second spring at University of Vermont, unfortunate timing to be pre-tenure, an undergrad joined my lab who was a microbiology student and very interested in poultry, and had a job at an agricultural supply store that sold chicks in Massachusetts. And I decided that maybe we should look at chicks, because I found a paper from the CDC that suggested that most illnesses were actually associated with chicks and not adults. I thought, well, that would make sense. And I made plans to recruit people to let me sample their chicks that they'd purchased. And then COVID, and the lab was shut down, the university was shut down, the student went home, and obviously we weren't going to go into people's houses to sample the chicks they had living in cardboard boxes in their bathtub or garage or whatever. So I thought that was going to be the end of that. And my student emails me from Massachusetts in like April, and she's like, hey, I think my boss would agree to let me sample our poultry at the store. Would you be interested in samples if I could get them? And there's only one answer when a student is like, hey, I want to do research. You're like, oh, okay, yeah, sure. So she ended up getting about 56 samples, kept them in her freezer. I don't know how her family agreed to let her do that. I got a few people locally to put out baggies or clean containers of soiled bedding from their chicks on their porch, and I'd go pick it up. But my husband was absolutely opposed to having any fecal material in our fridge or freezer, so I made a few unauthorized visits to the university to drop off samples, because my key card still worked. There was nobody around. We ended up getting 79 samples. She came back as a tech over the summer when we reopened, and a local egg store was like, hey, we had contacted them. They're like, yeah, you can come sample. And the 80th sample from turkey chicks was positive for salmonella. And we were so excited, which it's just such a weird food safety thing. Like, you're like, salmonella, we got it. And that really kept the project alive. And then we sampled one farm in the fall with adults, and we found our first adult sample. And so we just kept the project going.
A worldwide leader in animal nutrition, Adiceo's portfolio of products includes methionine, the full range of vitamins, enzymes, organic selenium, probiotics, mycotoxin management strategies, and palatability products. With such a diverse offering, Adiseo supports its customers with a broad range of expertise, tools, and services to help them maintain a competitive advantage. Adiseo, fueling predictable profits. To learn more, visit Adiseo at www.adiseo.com.
And your technique has been a traditional culture. Have you been doing any molecular techniques or?
We do for Campylobacter. We expanded into Campylobacter in 2022 when I had a good grad student who I felt like could manage that. But we've had no luck culturing Campylobacter. We can find it using the Hygiena-Bacs, so PCR-based methods. But despite talking to so many people who are experts at culturing Campylobacter, it's been really hit or miss for us.
I think it's a common experience as laboratories have difficulty getting Campy to grow. So it's a fastidious organism.
It is a nightmare. I sometimes think that people who can get Campy to grow have sold their soul for the ability. Or there are a million Campies in a trench coat or something. Because, they're always like, oh, yeah, it's so easy to get it to grow. And I'm like, yeah, you've been doing it for 20 years. Like,
yeah.
I'm sure it's easy for you.
So in terms of what you're finding there in Vermont, you think that the backyard population of poultry is not that much of a bacterial carriage? Or?
So after 2020, we started finding a lot more Campylobacter. We found our first positive in 2020. And then in 2021, I think 7 of the 13 samples we took, farms we sampled, were positive for Campylobacter or for Salmonella. It was a complete turnaround. Some of it was where we were sampling. So we sampled primarily in town. I'd say over 50% of our samples were from town in 2019. And as we started getting into more rural areas, we did start to find more Salmonella. It ended up overall not being statistically significant. But looking at the data from 2019 through 2021, with, I forget the exact number of farms, it was in the low 30s, we found overall 19% of flocks had Salmonella. And we moved away from cloacal sampling partially because of not being able to always sample all the chickens, because we'd be like, hey, can you please lock up your chickens? And we get there and they're running around in an open yard. And you're like, well, an hour later, like, maybe we should probably head home.
What samples did you go to? And if you didn't do cloacal samples, how did you sample?
So eventually we started doing bedding samples. And so we do a combo sample from like the nesting boxes and the area near the water or the area near the feeder. And, really filthy bedding. To be fair, people don't clean their houses that much. We've seen so many really disgusting coops, not always correlated with Salmonella presence, but definitely easier to get soiled bedding from.
Well, it does not go with Salmonella maybe being an intermittent shedder too. I mean, if you're there in a day, you may miss the shed.
Yeah, for sure.
Whereas an accumulated sample, you'll have more of an opportunity to find accumulated.
Yes. And the other tricky thing with cloacal swabs is if the chicken hasn't pooped recently, there's like nothing on your swab. You don't want to make the chicken bleed. So getting enough sample to really detect Salmonella is really hard.
I think in commercial poultry, I've understood that too, talked to the folks, particularly the primary breeders that really look for Salmonella hard. They start looking for accumulation points like fans, that have all the air go over it, and a boot swab where you walk the whole house, not just an individual bird.
Yeah.
Yeah. So it sounds like you're finding the same thing in the backyard flocks. But you think it's about a 20% or 19% carriage rates, what you found?
Yeah, that was from 2019 through 2020. More recently, the 2022 through 2024 sampling, we found 13%. So there is a little bit of variation. I had hoped we'd get enough sequence samples from 2021 that we could trace it back to, there was a wild bird epidemic of Salmonella typhimurium, and I was like, oh, maybe that's where it's all coming from, because wild birds like chicken coops too. There's food.
Right.
But It was a project where I had, I don't know, there were probably at least 2 dozen undergrads over the years who worked on it, and data management and isolate management was a challenge. So a few samples disappeared into the ether, shall we say. And so we don't have we didn't find any Salmonella typhimurium in the couple of samples from that year that we were able to sequence.
That's maybe a further question, but ever work with the animal owners, like the people? You ever do any sampling? Do they offer?
So I did have someone who was like, my husband keeps getting Campylobacter, which interestingly, she was insistent it wasn't from her birds. It was always from somewhere else. She's like, could you sample him? And I'm like, we are not, we are not authorized to take human clinical samples, so no.
Okay.
So no, we have not sampled the people.
And in terms of any type of genotype or any type of connection between the different samples, have you found anything? Same species of Salmonella or Typhimurium or?
Yeah, so we mostly, if you look at by isolate, it was mostly Schwarzengrund, but that was influenced by a larger flock that had a lot of Schwarzengrund. If we look at by farm, it's really more Enteritidis, which is interesting because it's also the primary serovar we find in the chicks that are being sold. So it suggests there might be some persistence from hatchling to adult. But then again, people buy birds all the time. Predation is a huge issue around here, and people do not do an all-in, all-out approach. So it could just be repeated reinfection for all we know.
Well, do you find different ages? You say Enteritidis in the babies, but do you find a shift in populate or type of Salmonella as a bird's age, or not? Not necessarily.
That's not something we've looked at. That's interesting. I have a postdoctoral researcher who's doing some genetic comparisons. And so far, it seems like by serovar, there's not a lot of differences between the Salmonella in the adults and the chicks. Like they'll be like right next to each other on a SNP tree, core genome SNP tree. But I'd say one thing we do find more of a little bit in the adults is a little bit more antimicrobial resistance. But that's also very serovar dependent. Like we do not find antimicrobial resistance in Enteritidis. And if you get into something like Schwarzengrund or Infantis or Kentucky, we find more. We don't find much Kentucky either.
Yeah, really? Huh. Do you find any history on the flocks that have been treated with antibiotics? Do the owners tell you, or?
So there are not that many veterinarians that'll treat flocks, backyard flocks. There are a handful in Vermont. But mostly people go for, we did a survey, a small scale survey, and then a larger survey early on. And most people try a combo approach, and natural remedies are pretty common. And from having sat on the Facebook pages, trying to get a feel for what's going on, definitely a lot of home remedies. It's expensive, and the chicken, unless it's a pet, is not usually worth the vet bill.
Sure, right.
Especially since most of these flocks are like maybe an average of 4 to 8 birds, depending if you're rural or urban.
So in terms of implication for the commercial poultry industry, I think that was something else too. The backyard flock, particularly in big chicken states down, the chicken belt down here, always consider backyard flocks as a nidus. Particularly say influenza these days and time, but also laryngotracheitis or and I don't know about salmonella. I mean, that's what you've been looking at. What do you think about backyard flocks as a reservoir?
Honestly, I don't know that I'd say they're a reservoir. I mean, I think it depends on a lot of things. If you're looking at pastured poultry, then yeah, backyard flocks in the air are probably going to be a bigger factor. But I would think if you have good biosecurity practices, it's maybe not so much of a deal for like indoor raised birds.
And I think that's always our hope too. I mean, it's been
I mean, you can't control the flies per se or the wild birds, but there's definitely less of a risk. And we actually have not seen that many cases of HPAI in the Vermont backyard poultry. A survey we did a few years ago suggested that like almost 25% of Vermonters had or currently had backyard poultry like in that year, the last year. And driving down the road,
Validates that.
that
Yeah.
rings true. But we only have had maybe a dozen flocks, I want to say, over the last few years. Like it's intermittent. It's not decimated the backyard poultry industry or flocks the same way. Not sure why on that.
Yeah. I think too, it's just not in the news right now. There's commercial flocks making the news, particularly the leghorns, but for influenza. But of course, salmonella is always a concern. And, the thought was back with, backyard birds being handled more frequently than even commercial birds, that would be an opportune source if folks aren't doing themselves good sanitation.
Yeah, I mean, Vermont, anytime HPAI is found, it makes the news. So, and we don't really have any major commercial flocks. There's a few pastured poultry producers. I don't think we have any major egg operations, but so yeah, I feel like it's pretty well known. Honestly, my sort of off the cuff hypothesis is that they get exposed to more of the regular avian influenza through the wild bird population, and so maybe there's some background immunity or something. I don't know if that makes sense, but they definitely haven't been decimated to the degree that one might expect given their access to the outdoors and wild birds and such. But then again, leghorns, and maybe there's a breed susceptibility thing. Vermonters do not generally buy leghorns. I've seen a handful, and that's it.
Yeah, I think it also has to do a lot with exposure. So not having a concentration there probably helps out too. So ongoing research, what you're working on now?
So we just submitted a manuscript to Zoonoses and Public Health on the adult poultry, looking at Salmonella rates, Campylobacter rates, risk factors, serovirus found, and antimicrobial resistance. And we're working on the same thing for the chicks. That project, we wrapped up sampling in 2023, and so we had 2 years of super intensive sampling at agricultural supply stores, and then a third year that was slightly less intense. And we're still waiting on a few sequences before we can completely wrap that up, but we're hoping to submit it this spring as well. And then comparison of adult and chick genomes and such.
And can you track, you say your commercial or I guess farm stores source chicks, but where they get their chicks from? Can you have the...
Yeah, we have some data on that. Basically, all the major hatcheries that sell to the backyard poultry people are located in the Midwest. About the farthest east you'll get is Pennsylvania. So most of them are Midwest or South. We did talk to the stores about like where they got them from. But even if they said, hey, I get my birds from hatchery X, then it turns out that, we compare the breeds that their sampling boxes from to the list of breeds the farm or the hatchery sells. And we're like, okay, you're definitely getting breeds from somewhere else as well. Cause,
Right.
this hatchery doesn't sell meat birds. So, okay. So that was a factor that was interesting. There was a lot of variation by hatchery in how much salmonella we found. There was one in the Midwest that first year we got samples from that, like 70% of them were positive for salmonella. And some of that was probably the store because they kept their chicks in these big rubber tubs. And it was, they get more chicks in as chicks were sold off, and then they're combining them in the tubs. And it was pretty dirty, and tubs were open to consumers. And those ones were really hot for salmonella. Like sometimes you'd get samples back and it looked like you had a puriculture, and you're like, okay, are we sure this isn't the positive control? It is not the positive control. The following year, we were able to get more of the direct shipping boxes from the hatcheries, and test those pads, and it was like 50%, but still pretty bad. On others, it was like less than 20% of the samples were coming back positive. So a lot of variation.
So all these years of sampling and looking and finding some maybe wisdom we can learn from your learning phases. Talk about maybe cloacal versus environmental samples, or maybe some other things you've learned along the
Yeah,
way.
I think environmental is definitely the way to go. Um, the thing that really shifted us from doing cloacal to environmental was reading a paper from Australia on backyard poultry, and they'd done kind of a composite sample look with manure. But when you've got bedding, it's like hard to separate the manure out. So that was why we went with bedding. Another thing with this sort of field research, it was really important to eventually develop like very standardized procedures. But it definitely went more smoothly when I had grad students who had responsibility for pieces who were very meticulous, as opposed to a bunch of undergrad researchers who could give 3 hours a week to the project. And you're like, please do X. And then they bring back the sample logs, and you're like, what does this mean? Because you can tell people what to do, but you can't be there all the time. And detangling the data was just a nightmare sometimes. And even working with stores, and you're like, okay, you told me, we'd call them in February or January, and they tell us when they were planning to have poultry. And we call them 2 weeks ahead of the date, and they're like, oh yeah, our poultry were here last week. And you're like, what? Yeah, they were gone. They're long gone. Or you'd call, there are some stores that got poultry in every week. You'd call and they're like, yeah, we have poultry. And you're like, please save the boxes. And you get there and the boxes are in the dumpster. You're like, well, I just drove an hour. Do I dumpster dive?
That sounds very common. Other life experiences too. I think in commercial poultry, I've been in facilities exactly that. Always a day late, dollar short, whatever that is. You show up and agreed upon time, and things have happened, and it's moved on already.
I think one of the big things I took away from the consumer aspect is, there are a lot of good suggestions from the CDC and the USDA, but none of it is enforced. So we had hoped originally to look at so backyard poultry is regulated under Subpart E of the National Poultry Improvement Plan, and short of avian influenza and Salmonella gallinarum pilarum, it's all voluntary. And so these hatcheries, it became a really big thing to say you're part of NPIP. But what they meant by part of NPIP was sort of a buyer beware thing. Ideally, for the consumer's sake, it should have been, hey, we're participating in salmonella monitoring. But sometimes they would claim NPIP just because they were part of the mandatory programs. And you're like, I don't think you can count that. Um, and even if they were actually participating in salmonella monitoring, like the hatchery we were finding 50 to 70% of the samples positive was ostensibly part of the salmonella monitoring plan, because it's run by the states and it's up to the state's discretion how to enforce it. Interestingly, I think HPAI really forced the hatchery's hands, because if you look at the human cases, for the first time in like a decade, they were under 500 last year. And that is unheard of.
It's the salmonella cases related to backyard poultry, is what you're saying.
Yeah, yeah. So I think then they had to actually be really cautious with biosecurity and really work on that.
I think too, we talk about, chickens and salmonella. That's I guess a long-term relationship.
Yeah.
The salmonella has lived well in chickens for millennia.
Oh, I know. Yeah. And the chickens, we tried to figure out if we could tell if the adults were sick with salmonella. And you have a chicken out there with a prolapsed vent and just looking like super ragged, and it'd be negative for salmonella back when we were doing cloacal swabs. And then a chicken that looked perfectly healthy would come back positive. Now, we can't prove that the one with the prolapsed vent didn't have salmonella 2 weeks ago and clear it and pass it on to the other, but because we did have that happen. The first farm where we found salmonella, the owner was like, "Okay, well, which chicken is it?" And I'm like, "Well, you had 5 barred rocks, and because we've never found salmonella, we weren't like labeling the barred rocks in any way. So we'll have to come back out and sample your barred rocks." And when we did that, none of them were positive.
Mhm.
So
On
she's
that
like, "What?"
day, that you
On that
sampled.
day.
Yeah, right. Yeah, that's a
And I
challenge
think
for
we had
sure.
sampled her coop as well. But yeah. So I don't know if the chicken had cleared it. I don't know if it just wasn't shedding that particular day, but
That sounds like a very common frustration in chasing salmonella. It's amoeboid. It moves around, and then when you think you've got one serovar controlled, then another one pops up, and
yeah.
that's been a commercial side's been challenging. So it's interesting to hear from the backyard poultry. I think I would have thought it'd been more positive, more prevalent than what you've reported.
I mean, honestly, in the adults, like ours is the highest rate of salmonella that anybody has found. Like it was so far outside the average of like 2 to 5% that I was worried when we published that data that somebody was gonna be like, "Are you sure you don't have lab contamination or something?" Um, but obviously we had safeguards in there for that. I do think some of the fact that we have so much rural poultry in Vermont and so many people who haven't had poultry long term plays a role, as well as like the constant wildlife contact and people constantly having to replenish their flock because a weasel
Right,
or a hawk or a
right.
bobcat or a raccoon or a bear ate a bunch of the birds, and so now they're buying 5 new chicks and adding them to the pile.
And we definitely know varmints, mammals, raccoons, possums, skunks carry bacteria. I think I've seen it a lot with Pastorella. So surely we think salmonella would ride along with them too.
Well, I mean, I feel like anytime you ask, "Does this animal carry salmonella?" the answer is yes.
There you go.
So
Maybe that's a good, maybe a good lesson to wrap up on in terms of salmonella and outside the commercial poultry, which is interesting for those of us that are in the commercial poultry. Kind of look across the fence and maybe point fingers at them, but then you got fingers pointing back at us too.
right. Yeah, I think one of the really interesting things too is, a lot of in Vermont, there's a lot of interest in introducing kids to agriculture through embryology projects or taking them to farms. And I'm always like, can you just like wait until they're at least in first grade? Because maybe taking a 3-year-old, having 3-year-olds hatching baby chicks isn't your best plan. Maybe taking a preschool class to a local farm to pet the chickens is not your best plan.
I do think I think you hit a nail on the head though too. Is about consumer education. Try to do the best we can. With the animal to limit the amount of pathogens, but then there's a responsibility. Whether it's consumption of food or handling an animal. As a veterinarian, when you wash your hands, pet your dog, wash your hands. And don't let your dog lick you across the face. That's not a good idea.
I've never understood that. I mean, I've seen dogs eat way too much poop.
There you go. You're right. Oh my. But I think there's some education there that's everybody gets frustrated with, and how to get to the masses.
I don't know. And trust in experts is so low. Like I've seen people on the Facebook pages be like, "Well, CDC says I shouldn't pet my chicken, so I'd better go out and kiss it."
Right.
I'm like, okay, if CDC said you should jump off a bridge, or shouldn't jump off a bridge, would you go jump off the bridge just to spite them? Because like they actually do have some knowledge.
Well, let's go ahead and wrap this up, Andrew. This has been a fun conversation. Okay, this is an area that I don't delve in. We've never met before. This is awesome.
Thank you.
It's time for our famous 3. One of AB Vista's core strategies is to give customers the flexibility to do more with less, which is a common theme among many companies and producers in today's industry. As a science-driven company, AB Vista has proven results to help our customers achieve optimal performance using customized programs with our core phytase and xylanase.
It's been a great conversation. Before we go, there's some other personal questions they'd like us to ask. And any resources or references in your professional, inside the poultry world or food safety that you've relied on through the years?
Definitely read a lot of applied and environmental microbiology papers and Journal of Food Protection papers, and I think Poultry Association journal papers. Actually, I think some of the papers from the 90s where they were investigating like persistence of salmonella in laying hens were really, really, really helpful. So, I'm a big fan of going back to the old literature because,
Okay, that's good to know. Stand on other people's shoulders. That's good.
yeah, people did a lot of really important research in the 90s, and they might not have had sequencing and fancy stuff, but it's solid and really important.
And another question, maybe a more personal question, outside the profession, is there a book or a reference or that you go to or lean on or prefer, a favorite book maybe you've read or?
I mean, I'm a Christian. In the Northeast, that's actually really rare, surprisingly rare, compared to say like the Bible Belt. But I do rely heavily on my faith and on the Bible. And beyond that, let me think.
That's a good place to stay. I mean,
Yeah,
I'm a fan of that too. Yeah,
yeah.
yeah, yeah.
I really like reading The Atlantic. Their science journalism is really, really good. So if you want to read good science journalism, they've got some good people and a nice educator discount and a
There you
variety
go.
of
Okay, a plug for
topics.
that.
Yeah.
And then one more question was counsel for those that are up and coming. Compared to me, you're relatively young, but even your folks in your lab and the folks that you're training, any counsel for them as they're pursuing their career in agriculture?
I think there is a lot of things that haven't been explored. And, it's hard to break into a newer area. There is a sense in which if somebody hasn't studied something, it's probably because it's either really hard or there's no funding in it. And USDA hatch funds, because we administer a lot of that as pilot funds, was a major factor in being able to explore this as much as I have. But really, those places where your background and your experience and your curiosity meet a hole in the research are really fun places to work. And so, I think the ability to explore some of those things is really one of the best parts of being a scientist.
So be inquisitive and be energetic is what I'm hearing. Yeah.
Yeah, and like even if it seems like it's a little out there, it's probably worth at least exploring a little bit.
There you go. That's good. Again, thank you so much,
You're welcome.
Andrew, for your time today. I really enjoyed this. I've learned some things. Like I said, I thought prevalence was much higher in the backyard block, so this is,
Yeah.
good information. Yeah.
Thank you. It's been a delight.